Hello and welcome to the course Social Behavior and the Brain, an introduction to social neuroscience. I am Dr. Arik Verma, an associate professor at the Department of Cognitive Science at IIT Kanpur. This is the final week of the course. We are talking about social affect processing and we are trying to understand the neural underpinnings of emotional regulation in this lecture. Now, in the previous lecture, we were discussing about the fact that verbalizing one's emotions and feelings, especially negative ones, actually may lead to a diminished experience of the same. So, when we talk about our negative feelings or we write about our negative feelings, it leads us to a slightly reduced intensity of that experience. It takes us to an automatic down regulation, which is very difficult to sort of understand that whether it is happening through intentional or unintentional emotional regulation and basically where these effects are coming from. But one thing is very clear that expressing our negative feelings, both either by writing or by speaking out or reflecting over them, actually has very well established physical and mental health benefits. Now, this disruption of negative affect has been shown to have physical and mental health benefits, as I just said, and has been utilized in several therapeutic approaches. In this lecture, we will try and understand the possible neural mechanism for how this is happening. Now, Lieberman proposes the area of the brain called the right ventrolateral prefrontal cortex as playing a central role in this neurocognitive mechanism that governs the said disruption of negative affect through the process of verbalization. The RVL-PFC, that is how we will refer to this from now onwards, the RVL-PFC has long been associated with inhibitory processes and has also been identified in a large number of studies examining the symbolic processing of affect. Now, what is this symbolic processing of affect? We will talk about this going forward also, but the idea is when you are verbalizing something, you are basically giving words. In the process of speech production, if you've done my course on psycholinguistics, when we talk about the fact is when you conceptualize, from the process of conceptualization to lexicalization, when you are putting pre-verbal thoughts into words, you are basically looking for words, you are concretizing something that is amorphous, that is just at operating at a phenomenal level, and once you are making that connection, it is reducing the intensity of those feelings. Remember what James said, what Benedict Spinoza said, and so many others. So, the right ventrolateral prefrontal cortex has actually been associated with a large number of studies which have been involved in examining the symbolic processing of affect. So, symbolically transforming into symbols what was just a physiological feeling or a cognitive feeling of sadness or anger or anything like that. Now, given the fact that the RVL-PFC is involved in both of these roles, inhibitory processes and regulation and symbolic processing of affect, according to Lieberman, this makes RVL-PFC as a real candidate for driving these disruption effects. As these disruption effects appear to derive from the symbolic processing of affect, which may be leading to the inhibition of affective processing. So, the idea that is being presented here is that it is we were trying to see whether intentional or unintentional emotional regulation is happening, but the hypothesis that is put forward by Lieberman is the fact that these negative effect or the disruption of this negative effect is happening due to the fact that we are symbolically processing affect. And how or what is helping us symbolically process affect is this right ventrolateral prefrontal cortex. This is basically the area that we are talking about here. So, you can see that there is a huge overlap between the areas for intentional regulation as well as the areas for affect labeling, and this has been found in several studies. So, there is a large number of studies that you can basically see have actually found the same kind of findings. Now, well, there seems to be an ongoing discussion about the neural regions that are involved in inhibitory processes. If you look at them, the prefrontal cortex as a whole, the dorsolateral prefrontal cortex, the anterior cingulate, the RVL, PFC, etc. There are so many of these areas of the frontal cortex that are involved in emotional regulation, all kinds of inhibitory processes. But typically, the right ventrolateral prefrontal cortex is something that comes up in anybody's imagination of what the important areas for inhibitory control are. So, several neuroimaging studies using, for example, the go-no-go task or the flanker task or the Stroop task have actually identified the right ventrolateral prefrontal cortex activations associated with trying to inhibit a prepotent motor response or trying to ignore task irrelevant information that would lead to an incorrect response. So, the role of the RVL-PFC in inhibition is very well established across different kinds of studies using different kinds of experimental paradigms. Also, these tasks have found that the RVL-PFC activity is linked with faster reaction times on inhibition trials. So, when you are able to successfully inhibit irrelevant responses, and for more successful inhibition trials than unsuccessful inhibition trials. Also, RVL-PFC activity has been found to be greater for harder inhibition trials. It seems that the area is putting in that much more effort than as compared to easy inhibition trials. So, if you look at these studies, it seems that this area is certainly involved in emotional regulation or inhibition of emotional or affective or task irrelevant information. Also, children with attentional deficit hyperactivity disorder typically show impaired behavioral performance on motor inhibition and also show less RVL-PFC activity during inhibition tasks than controls. So, there is some data, some of this data that is coming from patient studies as well. For instance, in one of the studies, the experimenters observed better motor inhibition in an ADHD sample after neurofeedback training, also correlated with increased RVL-PFC activity relative to a sample that did not receive such training and who did not receive, who did not report higher RVL-PFC activity. Also, studies which are documenting effects of permanent lesions or temporary lesions through TMS in the RVL-PFC have also led to impaired motor inhibition. So, the specific type of inhibition that the RVL-PFC seems to be linked with is motor inhibition, but again this may have other consequences as well. Also, pharmacological studies, when you use certain kinds of drugs, so pharmacological studies wherein participants receive serotonergic agonists, disrupt the effects of the serotonin drugs associated with enhanced self-control and diminished impulsivity, observed greater activity in RVL-PFC during motor inhibition trials. So, even in drug trials, basically when the drugs are affecting and they are increasing self-control or they are decreasing impulsivity, they are linked with, they are probably doing it through increasing the activity in the RVL-PFC. So, this is something which basically tells us that this area is very critical for inhibiting prepotent motor responses, for decreasing our impulsivity. Now, an interesting study by Goel and Dolan has proposed that RVL-PFC may be involved in non-motoric forms of inhibition as well, such as the inhibition of belief. Now, inhibition of belief is something more in the ballpark of emotions. Let us look at this study. In their study, participants were asked to assess the validity of syllogisms. For example, does the conclusion logically follow from the given premises? And that were either sound, so either the syllogism was correct, deriving from true premises, or they were incorrect, deriving from false premises. For example, participants had difficulty accurately identifying a valid syllogism as valid if it was unsound and therefore not true. So, this is interesting that, for example, the premise is all addictive things are expensive, some cigarettes are inexpensive. It is valid to conclude that some cigarettes are not addictive, although the first premise and the conclusion are both false. So, people were having this difficulty identifying a valid syllogism as valid if it was unsound and therefore not true. So, people were finding it difficult to do this task. Interestingly, during this task, the RVL-PFC was the only region of the brain that was found to be more active when participants actually overcame their belief bias and indicated that this kind of syllogism was valid. See, if you look at the previous example, they had to overcome their basic belief and their basic knowledge and tell that, oh, the syllogism is true because the premises are true and so on. So when they were supposed to do this, or when they were successfully being able to do this, it was the RVL PFC that was implicated. It was the RVL PFC that was showing greater activity as compared to the other regions of the frontal cortex. Also, a number of studies on active deception have also implicated the RVL PFC in this aspect of inhibition of belief. So, if you look at these studies, wherever individuals are required to inhibit what they knew to be true and then to say something as well, say for example, I tell you, oh, sun rises in the west. I know that this is false, but I am overcoming that and I am saying this. In these kind of scenarios, the RVL PFC was supposed to be recruited. Now, the RVL PFC has also been implicated therefore in the symbolic processing of effect, in a large number of studies. So, this symbolic processing of effect, again remember I just talked about this very shortly. Symbolic processing of effect is basically putting feelings to words. That's basically the feeling, basically the idea that we will work with. So, the SPA or the symbolic processing of effect refers to the explicit linguistic or propositional processing of one's own effect. I am feeling sad, I am feeling angry. When you say this, when you verbalize that, it sort of takes away the edge of how angry or how sad you are feeling, because you have concretized all of that into a sentence, and that sentence may be correct, incorrect, reasoned with, argued against, negotiated with, and so on. The effect of others, for example, she looks frightened, evaluatively valence categories, for example, terrorists are bad, or the value of response options. Say for example, I will lose my money if I keep my money in the Betamax stock or X stock or Y stock and so on. So, this aspect of symbolically processing effect seems to be recruiting the right ventrolateral prefrontal cortex that we are referring to by the acronym RVLPFC. Now, across a range of these studies, across a variety of these studies, RVL-PFC tends to be more active during symbolic processing of effect than the non-symbolic processing of effect conditions, particularly in the case of negatively valenced emotions or negatively valenced feelings. For example, Cunningham and colleagues presented participants with famous names like Bill Cosby and Adolf Hitler, who are generally viewed either very positively or very negatively. So, they basically presented these kind of names, and in this experiment what happened was that on some trials participants were asked to decide whether the target was alive or dead, but on some other trials they were asked to decide if the target was a good or a bad individual. So, on all trials, implicit effective responses to the targets were anyways affected. As soon as I take the name Adolf Hitler, some kind of value judgment will anyways creep up, even if you have to just say, oh, he is dead or he is alive, or even if you have to explicitly say he was good or bad. Okay, but explicit SPA would be expected only on trials, which I said when they were asking good or bad. Okay. Now, Cunningham and colleagues actually observed that the RVL-PFC along with the medial prefrontal cortex were more active during good-bad judgments than during alive-dead judgments, suggesting that these are the two regions which are involved in the symbolic processing of effect. Because you are propositionally, in this case you are propositionally processing these two individuals or any individuals in question. Now, a number of studies that have focused on explicit judgments about emotional aspects, oh, I am feeling sad, happy, angry and so on, and voices demonstrated greater RVL-PFC activations to emotional than to non-emotional judgments. Okay. So, when you are making emotional judgments, when you are doing this, basically what it leads to is leads to the recruitment of this particular region. A study that specifically compared negative emotion judgments to neutral and positive judgments observed greater RVL-PFC activations to negative judgments similarly as was found in the Cunningham study. So, it seems that while the RVL-PFC is certainly getting recruited in cases where symbolic processing of effect is required, it is all the more critical for cases where negative emotions are being processed, when negative feelings are being symbolically or propositionally being addressed. Several studies have also observed the fact that reading negatively valenced words, just read a list of negatively valenced words, things like anger, sadness, cruelty, gory, all of those kind of things. So, reading negatively valenced words is also associated with greater RVL-PFC activity than reading neutral or positive words. So, it seems that this region is certainly getting recruited every time you are encountering or coming across negatively valenced emotional information. Nomura and colleagues, for example, compared difficult emotion judgments to easy emotion judgments. Presumably, in their task, the difficult emotion judgment requires relatively more top-down elaboration of the emotional qualities of the stimulus than the easy judgments, and thus would involve more symbolic processing. In this study, participants were asked to judge the emotional expression or the gender of the target faces. Okay? So, either you tell whether the face is happy or sad, or you tell whether it is a male or a female. For half of the trials, the critical dimension was ambiguous. For example, half of the gender trials had faces that were ambiguous with respect to gender. So, it would require more detailed processing. Nomura and colleagues actually found that the RVL-PFC and the dorsal anterior cingulate cortex were the only regions of the brain that were more active during ambiguous trials than during unambiguous trials. So, it seems that some kind of critical processing is happening during in these regions. Importantly, however, the effect of the RVL-PFC was driven entirely by its response to ambiguous emotion trials, whereas the dorsal anterior cingulate cortex was equally responsive to both kinds of ambiguity. So, this is where the critical finding is, that when you are not very sure that the emotion is happy or sad or positive or negative, that is where the RVL-PFC is sort of getting recruited, and it is being asked to work, as compared to the dorsal anterior cingulate cortex is basically when responding to both kinds of ambiguity, gender ambiguity as well as feeling ambiguity. So, again, we cannot stress enough the importance of the RVL-PFC here with respect to processing, symbolically processing negative affect or negatively valenced emotions. So, a reasonable interpretation of these results could be that the RVL-PFC is getting recruited on an ambiguous emotion trial as participants engaged in explicit hypothesis testing about the emotional expression. Oh, whether it is sad, whether it is a happy feeling, whether it is a positive feeling, whether it is a negative feeling, you are basically trying to process that, and the processing is probably happening in this particular region of the brain known as the right ventrolateral prefrontal cortex, which again is consistent with the putative role that is being proposed in of this region in symbolic processing of affect. I have only this much in this lecture. I will see you in the next lecture, where we will continue our discussion about symbolic processing of affect and the role of the RVL-PFC. Thank you.